Hi everyone! Today we are going to be talking about the digestive system, which is chapter 41 in your book. We are going to take a little bit of a different approach than what your book lays out. We will start by discussing nutrition, and we're going to review this to a lot of stuff that we've learned, especially in Unit 1. And as we go through the digestive system, we've got a picture of a two-dimensional version of the gastrointestinal tract of a human right here. We are going to take a linear approach by discussing all of the processes that happen in the mouth, then in the stomach, small intestine, large intestine, through the anus. So we're going to start at one end and we are going to end at the other, just like the journey of our food. We are going to start with talking about digestion in humans and our anatomical structures, what processes occur at each location, what enzymes are involved, and what the functions of those enzymes are. And then towards the end of the lecture, we'll compare to other animals. So in the book, the comparative approach starts first and it ends with humans. So we're not going to go as in depth as the book. But we are going to focus mainly on the human anatomy and physiology of the digestive system. And with the comparative approach, we're going to review what we learned in Unit 4 with how digestive systems differ across various animals. All right. For the nutrition slide, we've just got some terms. Nutrition is the process of taking in, taking apart, and taking up nutrients from food. So taking in food is ingestion, taking food apart is digestion, taking up the nutrients into the bloodstream is absorption. And then we're going to have elimination, which is taking it out. So all of these processes occur in our gastrointestinal tract, which I'll often shorten as GI tract, or digestive tract, or digestive system. You also have seen in Unit 4 us refer to the gastrointestinal tract as the alimentary canal. But we essentially have one end, that's the mouth, a big long tube, maybe a stomach along the way, and then another end that is the anus. Right. And we can classify animals on what type of nutrition they ingest by distinguishing between plants and animals. So an herbivore is an animal that only ingests and gets their nutrition from plant, fungal, or non-animal tissues. Carnivores obtain their nutrition from animal tissues only. And omnivores obtain their nutrition from both animal and plant tissues or fungal tissues. All right, so nutrition provides animals with an energy source, right? So animals, in terms of where they get their energy, since we get our energy from organic chemicals, we are chemotrophs. Nutrition also gives us our carbon, and since we don't make our own carbon, we get our carbon from other organic material. We are heterotrophs. And we get other essential nutrients as well, not just our carbon and energy from our nutrition or our diet. So I've got one more slide on essential nutrients. Essential nutrients are elements or other molecules and compounds that are required from an external source. So they are chemicals that we cannot make on our own. So this includes the 6 elements that are most abundant in cells. And this is a review from Unit 1. You should know which macromolecules we find carbon, hydrogen, and oxygen. Hint, it's all 4 classes. And then what macromolecules do we find phosphorus in? Where do we find nitrogen? And then in which class of macromolecules do we find sulfur? So for example, we need sulfur in our diets because sulfur is found in proteins. So we need sulfur to build our proteins, but we don't need sulfur for our nucleic acids or our lipids or our carbohydrates. So sulfur is an element found in proteins only. In our diets, we also need amino acids. There are 8 essential amino acids. Now, all 20 amino acids are essential for living organisms in general, but out of the 20 amino acids, we can build 12 of them from their building blocks or from the elemental components. There are 8 amino acids that we cannot synthesize by our own anabolic pathways, and that means we must get them through our diet. So if you are eating a diet that has a complete source of amino acids, it means it has all 8 of these essential amino acids. So examples of those essential amino acids include tryptophan. Tryptophan is known to be the part of protein in turkey that makes you sleepy on Thanksgiving. They always say you get sleepy after a big turkey meal because of the tryptophan. I'm not sure if that's actually true, but it helps me remember that tryptophan is an amino acid. Lysine and methionine are also examples of essential amino acids. There are also certain fatty acids, some membrane lipids that we cannot make on our own. So there are certain fatty acids that are essential that we get in our diet. We need to intake vitamins and minerals from an external source, not necessarily through supplements, but through our food. So for example, certain foods are rich in vitamin D, which is really important in the wintertime. Sometimes we can synthesize our own vitamin D when exposed to sunlight, but in the cold, gray winter months, we need to have our vitamin D from an external source. And what vitamins are coenzymes. Coenzymes refer to an organic molecule or something that's required for an enzyme to function properly. And so vitamin D is important in a lot of cellular processes and a lot of enzymes. There are water-soluble vitamins and there are fat-soluble vitamins. Water-soluble vitamins include things like vitamin C. And with water-soluble vitamins, because they are soluble in water, if we get too much of them, we can excrete the excess in our urinary tract. In our urine through the excretory system. But our fat-soluble vitamins, an example would be vitamin E. These are not soluble in water. They're going to be hydrophobic, so they would have a lot of nonpolar covalent bonds. Our fat-soluble vitamins stay with us. They aren't excreted in our urine, so excess amounts can be very dangerous because they get stored in our adipose tissue, and if they reach a high enough concentration, they can become toxic. So it's important when you are taking vitamins, especially if they are supplements, it's okay to take an excess of water-soluble vitamins, but for our fat-soluble vitamins like vitamin E, make sure you don't take over 100% of your daily recommended dose. Okay. And minerals are similar to vitamins, except they can be inorganic. So for example, and they can be a part of proteins or tissues, not necessarily just enzymes. So calcium is a part of our bone tissue. Cofactors might include things like iron. Iron is a cofactor in our hemoglobin protein. So it's an external inorganic molecule and it's required for the functioning of that hemoglobin. And so cofactors might not necessarily function to help enzymes, but it could be other proteins as well. So vitamins and minerals help our proteins function properly, and some minerals can be components of tissues. All right, here's a review question from Unit 1. Let's look at these chemical structures. Which one is going to be water soluble and which one is going to be fat soluble? Now you may recall in the previous slide I told you the answer, but if I hadn't told you the answer, how can you tell? You can tell by looking at the functional groups. This vitamin right here is chock full of hydroxyl groups. And what we know about hydroxyl groups is that they are polar, and this will contribute to a more hydrophilic property. So things with polar covalent bonds are more likely to be hydrophilic. Here we've got one hydroxyl group, but then for the most part, we've got a long tail of mostly carbon and hydrogen. And these types of bonds, carbon-carbon and carbon-hydrogen bonds, tend to be nonpolar. And when we see a lot of nonpolar covalent bonds, then we know our substance is going to be more likely to be hydrophobic. So something that's hydrophobic, if it's not soluble in water, it'll be soluble in fat. So a fat-soluble vitamin is one that will be hydrophobic due to nonpolar covalent bonds. All right, now we're done with nutrition and we're going to move on to the major part of this lecture, which is the digestive system. As we go through, we're going to go through 4 processes of the digestive system, and I'm going to color code these in terms of where they occur. These are the major processes, but as we get into digestion, we are going to talk about all sorts of different types of enzymatic digestion and a couple of types of chemical digestion. I'd like to bring up the activity that I have for you to help you put all of this information together. I have listed a number of different processes, and on the second page of your handout, you are to label where these processes occur. Now, some will happen in more than one location. For example, mechanical digestion will occur in both the mouth and the stomach. So you can label mechanical digestion in both locations. All right. Now, some of our processes are going to involve enzymes or chemicals. For this purpose, I've put an asterisk next to these processes. And when you see an enzyme down here, you're going to match it with the process that it is involved in. So amylase, for example, digests amylose, which is a carbohydrate. So if it's salivary, that means it's released by the salivary glands. So in the mouth, you'll have a label for carbohydrate digestion, and underneath that label, you can include the sublabel of salivary amylase. So obviously salivary amylase is going to be produced by the salivary glands, but anytime you have a gland secreting enzymes, it's going to be squeezed into the digestive tract. So the digestion is actually going to occur in the mouth, the stomach, and the small intestine. Everything else attached to it is called an accessory organ or an accessory gland because it secretes stuff into the spaces along the GI tract. Okay, so ingestion is our first process. Let's color code this. Ingestion is eating, right? So taking in food. And ingestion is going to occur in the mouth. Our next process is digestion. Digestion is the major one, and we are going to get digestion starting in the mouth, continuing in the stomach, and also in the small intestine, but not in the large intestine. We will see several different types of digestion along the way. Mechanical will include churning and chomping and physically breaking down. So we'll see that in 2 locations. Enzymatic digestion, we're going to have 4 categories for each of the 4 classes of macromolecules. So we're going to see enzymatic digestion of our carbohydrates, of our lipids, of our proteins, and of our nucleic acids. Now, we don't think about our diets being very high in DNA and RNA, but all of the food we eat comes from living organisms, whether it be plant, fungi, or animals. And so if our food comes from other living organisms, then it will contain all 4 classes of macromolecules, even nucleic acids. And then finally, chemical digestion. We're going to see some with proteins in our stomach and we'll see fat emulsification using bile in the liver. So I'll give you 2 examples of this, and you'll know where those occur. Absorption means that once the food is broken down into their tiny little parts, the food is going to next be absorbed into the bloodstream. So absorption is when the breakdown products go into the bloodstream, and absorption will include absorption of nutrients as well as water. A lot of water is secreted in the digestive system, and if we don't get it back into the bloodstream, we will have massive diarrhea and die of dehydration. So absorption means taking up nutrients and water back into the bloodstream. So that will occur in the small intestine after digestion, as well as in the large intestine. And then finally, elimination is the removal of digestive waste. Let's see if I can, nope, we don't have any brown color. I'll use green. Elimination is the removal of our digestive waste. Our metabolic waste comes from metabolism end products in our cells, and that gets carried through our bloodstream and excreted by our kidneys and our excretory system. The products of digestion, however, is feces. So our digestive waste is feces or fecal matter, and this is going to come out of the other end of the digestive tract, which is the anus. So that will be where elimination occurs. All right. And again, we are going to take a linear approach from the mouth down to the anus, and we are going to discuss these processes as they occur. All right, so from this point moving on, I have a key or a code that I'd like to share with you. Anytime we have a process, let me go back to my purple. Anytime we have a new process, I'm going to underline it. So processes you'll find underlined. For example, ingestion, digestion, and mechanical versus enzymatic. So all of our processes are underlined. Our anatomical structures are in bold. I don't know how to write in bold, so I'm just going to say they are in bold. Oh, don't want to underline it. Our bold font, right? So here we go. And our enzymes or chemicals are going to be in italics. All right. So as you all are making your own study guides, I know I have a brief one available to you, but make sure you're familiar with all terminology that I use in my lecture notes, not just the ones in bold. The ones in bold refer only to the anatomical structures. Okay, so again, ingestion occurs in the mouth, which is called the oral cavity. This is also where our first stage of digestion occurs. So we have 2 types of digestion in the oral cavity. The first is mechanical digestion, which is just chewing. That's pretty obvious. So our food gets chewed up into smaller pieces because that begins the process of breaking it down. It is a digestive process. We also have an enzymatic digestion, but of only carbohydrates. We have an enzyme that is secreted from our saliva called salivary amylase. So amylase breaks down amylose, which is a type of starch. So we have enzymatic digestion, but of carbohydrates only. So we are going to get some complex polysaccharides being broken down into smaller polysaccharides or maybe even disaccharides. So we'll get the bonds broken between the monomers. So we'll break down those glycosidic linkages that hold polysaccharides together, and they'll be broken down into smaller poly or even disaccharides. So I always think about how a cheese puff dissolves in my mouth when I eat a cheese puff. It's a wonderful sensation. And that dissolving that we feel when we eat a cheese puff are the carbohydrates or the starch in the cheese puff being broken down into something smaller. We have secretion of saliva. So the saliva provides the salivary amylase, and the saliva also helps to lubricate our food and form something called a bolus, which is just that large glob of food that we swallow. And right when we swallow, I suppose swallowing is a process, so we could underline that, but I promise I won't test you over it. When we swallow, our epiglottis is a tissue layer that will block the food from going into the trachea. So our food is going to go down the esophagus to the stomach. It will not go into the trachea, which is a part of the respiratory tract that goes to the lungs. We'll learn about that next week. All right, so once we swallow our food, it moves through the esophagus into the stomach. And coughing will occur when that epiglottis reflux fails. So if you ever cough after drinking liquid, it's because a little liquid got through into the wrong pipe because of a failed reflux of our epiglottis. And so the food moves through the esophagus to the stomach by muscle contractions. So we have muscles lining the entire gastrointestinal tract. Just to review from our tissues lecture, the muscle is smooth muscle tissue, right? It's an involuntary muscle contraction that moves our food down to the stomach. So it's not just by gravity. So even if we're laying down on our stomach, on our backs, food will still move through our GI tract because of the smooth muscle contractions moving things along. And that smooth muscle contraction is called peristalsis. The food will enter the stomach through one sphincter called a cardiac sphincter. You don't need to know that for the test. And in the stomach, we're going to have some other processes. I'd like to focus on the chemical and the enzymatic digestion first. So food moves into the stomach, and what we know about the stomach already is that it has a very low pH. So it's a very acidic. So review, that means it has a very high proton concentration or a very low pH, but a very high proton concentration. All right, and so it gets this acidity from the cells that are lining the stomach. So we have epithelial. If this is the inside of our stomach, then these are the pits right here that are exposed to the inside of the stomach. All right, so we've got these, each pit goes down a deep distance and it's lined with these epithelial cells that function in secretion. So these epithelial cells are the simple columnar epithelium, right? They're simple, they're one row of column-shaped cells, and they're gonna excrete substances. So our chief cells excrete an inactive enzyme called pepsinogen. Chief cells secrete pepsinogen. And by itself, this is an inactive enzyme. All right. Then we have parietal cells that secrete hydrochloric acid. It uses a proton pump to push protons into this space, and it also pumps out chloride ions. The protons and chloride ions form hydrochloric acid, which is what makes the stomach so acidic. So this does two things. The first thing is that the hydrochloric acid starts to denature proteins. So protein denaturation is when the quaternary and tertiary structure and secondary structure comes apart and the proteins start unfolding. So this protein denaturation would be an example of chemical digestion. Chemical because hydrochloric acid is a chemical. So we'd say protein digestion begins with chemical digestion in the stomach. So it's only going to be of proteins. Okay. All right, the hydrochloric acid does another thing. Pepsinogen is inactive at a high pH, but at a very acidic pH, it folds into its proper shape. So the optimal pH of this enzyme is actually between pH 2 and 3. So pepsin is active only when hydrochloric acid is being produced. So we have pepsin enzymatically digesting proteins. So a protease is just an enzyme that breaks down proteins. Okay, so pepsin is secreted in the stomach as an inactive form, but it's activated in the presence of hydrochloric acid. So we are going to get enzymatic digestion in the stomach by pepsin, and that is going to help the enzymatic digestion of proteins or polypeptides into smaller peptides. Okay, so the stomach has a few other functions. One is it stores food. Food doesn't just pass through the stomach, it holds a lot and it's squirted into small little bits into the small intestine. And the sphincters, the cardiac and the pyloric sphincter, control the movement of food in and out of the stomach. There are skeletal muscles also. There are muscles lining the surface of the stomach. And these muscles contract and churn the food up and helps to contribute to mechanical digestion. So in the stomach, we have enzymatic digestion by pepsin, we have chemical digestion by hydrochloric acid, and we have mechanical digestion because of the muscle contractions of the stomach. And because of the acidity of the stomach, it's also useful to note that we have a lot of mucus secretion from those simple columnar epithelial cells, and the mucus secretion helps protect the lining of the stomach. If you have a gastric ulcer, that means you have a lesion in that lining, and that lesion is caused by an infection of Helicobacter pylori, which is a bacterium. Okay, we're going to move on now to the stomach. The small intestine. The small intestine is going to function in both digestion and absorption. It is the longest part of the gastrointestinal tract. There are 3 parts: the duodenum, the jejunum, and the ileum. We are going to get enzymatic digestion mostly in the duodenum, and we are going to get absorption in the jejunum and the ileum. So the absorption will be of both nutrients and water. And that absorption refers to the nutrients and the water getting back into our bloodstream. All right, so right here at the duodenum, this is where our liver and our pancreas secrete stuff right here. So we are going to talk about how the liver and the pancreas contribute to enzymatic digestion. All right, so starting with the liver. Our liver is the organ that produces bile or bile acids. These bile acids surround fats or triglycerides and emulsify them. Emulsification means the solubilization of a fat in water by the formation of a fat droplet. So here's a picture of oil and water. If we take these oily triglycerides and we coat them with an amphipathic molecule, that's hydrophilic on the outside, then we can actually mix that oil and water. In our digestive system, we're going to coat the lipids that we ingest through our diet, or the fats we ingest through our diet, with these bile salts. And they are hydrophobic on one side, hydrophilic or ionic on the other side. And so now these fat droplets can be soluble in the water inside the lumen of the small intestine. So the liver synthesizes bile, and our gallbladder stores that bile and releases it into the duodenum as the food is coming from the stomach. The pancreas will have two important functions. One function is to secrete bicarbonate, which is our body's natural buffer. So this will neutralize that acidic food coming from the stomach and helps buffer the pH back to neutral in the duodenum. All right, and then the pancreas also secretes a number of digestive enzymes. So digestive enzymes are produced by the pancreas as well as by the small intestine cells themselves. And together, the enzymes of the pancreas and the small intestine will complete the digestion of carbohydrates, proteins, nucleic acids, and fats. So we're going to finish up our digestion in the small intestine. And here are all the enzymes that are involved. We've already talked about the mouth. So with the mouth, we had salivary amylase, which is involved in carbohydrate digestion. Then we've got our smaller polysaccharides and our disaccharides moving through the stomach. We don't have carbohydrate digestion occurring in the stomach, but we do have proteins beginning to be broken down by pepsin. All right, so we've begun carbohydrate and protein digestion. When the food now enters our small intestine, we have enzymes being secreted from the pancreas and from the epithelial cells of the small intestine. The pancreatic amylases will further break down our polysaccharides into disaccharides, and then disaccharides will be broken down into monosaccharides. So if you see the word disaccharidase, that means it's an enzyme that breaks down disaccharides, and amylases break down polysaccharides. All right, so when we're done with carbs in the small intestine, monosaccharides are the end product of digestion. And then those monosaccharides will get absorbed into the bloodstream. So I'm going to square all of our end products of digestion. Protein digestion also completes in the small intestine. Smaller peptides get broken down into even smaller peptides, and then those peptides are eventually broken down into their basic monomer subunits, the amino acids. So amino acids are the monomers of proteins, and they are the end product of protein digestion. The names of the enzymes involved in protein digestion are going to be called peptidases or proteases. So if you see the word peptidase, know that it's breaking down peptides or proteins. If you see the word trypsin or chymotrypsin, again, I think of that amino acid called tryptophan, and I think anything with that prefix trip is going to be involved in protein digestion. All right, and now we have nucleic acid digestion occurring in the small intestine. Nucleic acids are first broken down into their nucleotides, and then nucleotides are even further broken down, and our end products of nucleic acid digestion are the 3 parts of a nucleotide, which includes the nitrogenous base, the phosphate group, and the 5-carbon sugar of either DNA or RNA, and that's what will get absorbed into the bloodstream. Our fat digestion involves the breakdown of a triglyceride, so we've got a glycerol molecule and hydrocarbon tails. We have lipases that break the bonds between the glycerol and the fatty acid tail, and our end products of fat digestion are going to be glycerol, fatty acids, and a monoglyceride is when we have a glycerol molecule with just one fatty acid attached to it. So our fats don't fully get broken down in the digestive tract. They get partially broken down and they're going to recombine again later on. And we'll talk about that in a little bit. Okay. All right, so in the small intestine, once the digestion is complete, the nutrients that are water soluble, the amino acids, the nitrogenous bases, the phosphate group, the 5-carbon sugars, and the monosaccharides, all of those water-soluble nutrients will get absorbed into the bloodstream. Once nutrients are absorbed, so they go from the lumen into capillaries, then they'll get carried by a vein called the hepatic portal vein. The hepatic portal vein means it's going to the liver, so it's transporting blood to the liver. And the nutrients get carried to the liver. Some metabolism will occur in the liver. For example, fructose might get converted to glucose, and then those nutrients will continue moving throughout the body. From the liver, it'll go next to the heart, and from the heart, those nutrients will be pumped throughout the body. So since these nutrients are water soluble, we're also going to have the absorption of water as well in the small intestine, and the absorption will occur in the jejunum and the ileum, which are the second and the third portions of the small intestine. And the small intestine is a good example of how surface area maximizes diffusion. The surface area of the small intestine is increased in 2 ways. The first is the folding of the epithelial cells, and these folds are called the villi. The microvilli are folds at the surface of the cell that you see right here. So there is a number of foldings that occur to help maximize the surface area and maximize the absorption of all of the nutrients that come from the digestion of our food. Now, fat molecules are absorbed differently, and they take a different route throughout the body. Remember that when we broke down our triglycerides, we got some glycerol, which actually is a water-soluble nutrient, so that's going to get absorbed into the bloodstream. But our fatty acids and our monoglycerides are going to be absorbed into the cells. They're going to be recombined back into triglycerides. So we're putting back together what we've already broken down. And then the triglycerides in our cells are coated with cholesterol and proteins to form chylomicrons. So the chylomicrons, you consider the cholesterol-coated fats that get carried around our body. The chylomicrons are not going to go straight to the bloodstream. Instead, they're going to go into the lymphatic system. So a lacteal is a lymphatic system vessel that will carry the chylomicrons to the lymphatic vessels, which go through the lymph nodes, and they end up being returned to the circulatory system through the lymphatic system. So the take-home message of this slide is that fat molecules are absorbed differently because of their insoluble nature. They are absorbed, they're coated with something to make it water soluble, and then they're carried through the lymphatic system before they return to the circulatory system. In the large intestine, digestion has occurred to completion. And so now all that's left is to absorb all of the water, and then microbes are going to just have at the undigested material. So the large intestine includes everything from the cecum, the colon, the rectum, and the anus. The colon is the major part of the large intestine where most water absorption occurs. So water absorption is the primary function in the large intestine. We also get the formation of our fecal matter. The cecum is going to be the first part, and this is going to be more pronounced in herbivores or animals that eat a lot of cellulose or plant tissues as part of their diets. The large intestine is the most dense bioreactor in our bodies. It is home to a thriving community of anaerobic microbes. So our anaerobes will grow by either fermentation or anaerobic respiration. You should review what these two processes are. And the fermentation that our colon microbes do not produce very good smelling fermentation end products. They produce things that are called volatile fatty acids. So this isn't nice smelling like lactic acid or ethanol. We get the formation of butyric acid or butyrate, propionate, and they smell really, really bad. Methane can be formed from anaerobic respiration. We also could get some sulfide from anaerobic respiration, which can contribute to a rotten egg smell. Oddly enough, methane doesn't smell. It sounds like it does, but it's fairly odorless. And so if you are wondering why feces smell bad or why farts smell, it is because of the volatile fatty acids and hydrogen sulfide produced by anaerobes in our colon. The cecum will have an important role in herbivores. Plant matter will be digested. Some microorganisms, some fermenting organisms will have cellulitic enzymes, or enzymes to break down cellulose, that humans don't have. So, fun fact, humans cannot digest cellulose on their own. We have a very small cecum, and cellulose digestion is not an important part of our nutrition. In fact, we consider cellulose to be dietary fiber because it just passes through our GI tract because of our small cecum. And we don't have the enzymes ourselves to break down cellulose or those beta-glycosidic linkages that are found in the cellulose polymers. Animals that do have a large cecum can absorb some of those nutrients from cellulose breakdown through the epithelial linings of their small intestine. All right, so here's a summary of everything we just learned about the digestive tract, starting with the mouth and then ending with the anus. This is a useful guide. I'm not going to repeat everything we just covered, but this is a useful guide for you all to take notes on or to use as you fill out your activity that I had given you. A brief comparison to other animals. We learned that not all animals have a complete digestive tract or an alimentary canal. In fact, some animals don't even have a digestive system. In sponges or in the phylum Porifera, food particles are ingested directly by choanocytes or collar cells through phagocytosis, and then the digestion occurs intracellularly. In comb jellies, which is the phylum Ctenophora, in cnidarians, and also in platyhelminthes, the free-living platyhelminthes anyway, they have a gastrovascular cavity with only a single opening called the mouth/anus. And for those that do have a complete digestive tract from a mouth to an anus, we have a variety of different digestive compartments along the way. So humans have a stomach, a small intestine, and a large intestine. Earthworms or annelids have a crop and a gizzard and a digestive tube. Insects, which are arthropods, have a foregut, a midgut, and a hindgut. Birds also have a crop and a gizzard as well as a stomach. And we're also going to talk about how mammals called ruminants have a specialized stomach called a rumen. So the ruminants is not going to be reviewed. We're going to cover that right now. The rumen is a specialized stomach found in deer and cattle. And the rumen is populated by anaerobic organisms that do cellulose breakdown and fermentation. So here's what happens in the rumen. First, cellulose is broken down by cellulitic bacteria. These are usually anaerobes that break down those bonds in between the monomers of cellulose. And then we get our glucose monomers after the activity of the cellulitic bacteria. Many of these cellulitic bacteria can use glucose as an energy source, and then we get fermentation. So when glucose is fermented, remember fermentation is an anaerobic process that results in the partial breakdown of an organic substrate. And so lactic acid fermentation was one type of fermentation we learned about, but in the rumen of a ruminant, we'll also get the formation of a whole bunch of other fatty acids. Again, a lot of them smell terrible. So we get these mixed organic acids as end products from fermentation. And then after the cellulitic bacteria and fermenting bacteria go to work, the cattle regurgitate their food and eat it again. And then these are the nutrients that can be absorbed and used by the animal. All right, that is it for the digestive system lecture. Next week, we'll be going over the circulatory, immune, and respiratory systems. Until then, take care and have a great weekend.